Hi, my name is Kamilah Islam. I was chosen this summer to work for the NIH SenNet CUSP internship at the University of Minnesota in the Institute on the Biology of Aging and Metabolism. So, a little bit about me. I'm a current junior at the University of Wisconsin Madison. I'm studying biology on a pre-med track. I'm hoping to become an obstetric anesthesiologist to help wage the major inequities in the healthcare system and access to healthcare specifically for those in marginalized communities and for people of color. This summer, I worked in the Niedenhofer lab. So I worked under Laura Niedenhofer and primarily worked with Beth Thompson, the project manager for the SenNet HUMIC grant. I worked alongside Kelsey Sobek and Sam Peters, as well as many assays and protocols. So here's a little bit about the structure of the Human U54 grant at the University of Minnesota. So it consists of four different departments. So first we have the administrative core, which is mostly just like my lab PI and other administration. Then we have the biospecimen core, which performs all the surgeries and gives us the samples that are then processed. And then I worked in the biological analysis core, in which we take the samples and perform various experiments. And then we send the data from that to the data analysis core, who then creates graphs and stats about and comes up with conclusions from the samples that we gave to them. So the overall purpose of SenNet is to study the senescent burden as we currently understand it. So senescence, as we currently know it, can be found throughout the body. So it can have good or bad functions in the body as well. So what we are studying specifically is chronic senescence and increased senescent burden with aging, which are linked to chronic illnesses and aging phenotypes. So the research questions that we're looking at specifically include where senescent cells are found, which cells are actually becoming senescent, and how exactly to identify senescence and what biomarkers can be used to identify senescence in different tissues. So human tissue processing, for this project, we studied both liver and adipose tissue. Most samples that we have are obtained from bariatric surgery patients and then sent to BioNet, which processes and distributes the samples. The amount and quality of the samples can affect the ability to use tissues in different experiments and the actual success of an experiment. So shown on this slide are the samples that we processed and for both liver and adipose and the basic demographics, so age range, BMI, things like that. And when studying adipose tissue, we're looking at both visceral and subcutaneous tissue. So visceral is the fat that is deep in your abdominal cavity surrounding your organs, while subcutaneous fat is stored right below your skin. So subcutaneous fat is more protective and you lose more of it as you age, while visceral fat increases with age and is associated with metabolic dysfunction and inflammation. So one protocol that I performed this summer was the SA-bgal assay, in which we took adipose tissue, both visceral and subcutaneous tissue, fixed it for 15 minutes, then we created an SA-bgal solution to incubate the tissue overnight. After that, we washed and imaged the tissue. The purpose of this assay is to identify the degree of senescent burden, and the higher accumulation of senescence shows in a darker stain of tissue, so more blue or green shade. So from this specific assay, we can see that there is an increased cellular senescent burden in subcutaneous adipose than there is in visceral adipose. The next assay I performed was a qPCR, in which we measured gene expression for several senescence-associated genes. So I isolated RNA from adipose tissue and performed the reverse transcription reaction and qPCR to quantify the expression of the genes, including P16, P21, IGFB3, CCL2, SERPIN2, and TNF. The results of the qPCR are in line with the results of the SA-bgal results, since we can see that in most all of the markers of senescence, that there is a increased senescent burden in subcutaneous tissue than there is in visceral tissue. So for single scope methods, we use two different methods. So for adipose, we use the parse nuclei isolation kit. So we first isolate the nuclei, perform the parse single-cell nuclei RNA-seq protocol, and then we send those results to the UMGC, the University of Minnesota Genomics Center, to perform sequencing of the data. So for adipose, we do single nuclei because dissociation loses about 90% of adipocytes, which is the primary cell type in adipose. And although nuclei isolation is still favored, the downside is that only 10% of the RNA is actually in the nuclei. And then for liver, we use a 10x fixed dissociation kit. And we first put the samples in fixative, then perform the single cell RNA-seq protocol. Then the UMGC does both processing and sequencing. So for the liver tissue, we do a fixed dissociation on liver because it only loses about 40 to 60% of hepatocytes, which is the primary cell type in liver. And fixing samples, although it causes loss, makes them more stable and easier to work with as well. So we can do more with low quality samples. And the difference between the protocols is that for adipose, we're doing single nuclei, and for liver, we're doing a single cell method. For the single nuclei RNA-seq experiment, I prepared the nuclei samples and performed the single RNA-seq experiment. The University of Minnesota Genomics Center performed the sequencing, and Samuel Peters did the bioinformatics analysis. On this slide, we have cluster analysis used to identify different cell types in the adipose tissue. We're focused on two different subjects, one being labeled as 1524, as you can see on the left, which is a 64-year-old, and the other being 1665, which is a 27-year-old. How cluster analysis works is that each nuclei is plotted based on its gene expression, and the cells with similar gene expression will cluster together, and cell types are assigned to the clusters based on the gene expression. So on the right, we're looking at sample 1665, both subcutaneous and visceral, and we can see that in each type of tissue, there is a different composition of cell types. For example, subcutaneous tissue shows a higher composition of endothelial cells, while visceral tissue shows a higher quantity of mesothelial cells. On this slide, we can see the degree of P21 expression detected in subcutaneous and young adipose. So how the data works is that the larger the circle, the higher the quantities of the cells expressed is, and the darker the color of the circle indicates a higher level of expression. So CDK and 1A or P21 is highly expressed in subcutaneous tissue compared to visceral tissue, and P21 is a consistent biomarker of senescence. So this is in line with the data that we've seen in the SA-bgal and the qPCR. The surprising conclusion that we came to from this is that P21 is also highly expressed in younger patients versus older patients, as depicted by the dark red circle next to young. Overall, we see a high P21 expression in young subjects and subcutaneous adipose tissue. On this slide, we have more results of senescent burden based on cell type. So in adipocytes, adipose progenitor cells, endothelial cells, and macrophages, we see a higher expression of P21 in young compared to old patients. In NK cells, there's a higher expression of P21 in the older subject, and T cells are the only cell type where there's a higher expression of P16 in the older patient versus P21. So the conclusions that I came throughout this process was that multiple assays should be used to identify senescence in tissue. SA-bgal can identify overall cellular senescent burden of a tissue. qPCR can be used to measure expression of senescent markers in tissue, and single-cell RNA sequencing can identify and characterize the specific cell types in the tissue that are senescent. Subcutaneous adipose has increased senescence to visceral adipose depots. P21 is surprisingly unregulated in young adipose, but it is cell type specific. In closing, I would just like to thank the University of Minnesota and the IBAM administration for taking me on this summer, as well as Beth Thompson, Samuel Peters, and Kelsey Sobek, as well as everybody in the Nadenhofer lab, because this opportunity would not have been the same without them. And I would also like to thank NIH SenNet Consortium for giving me this opportunity this summer. Thank you.